Tagging of endogenous RAD21 with an AuxinInducible Degron allows for rapid, near-complete cohesion loss. In HCT116RAD21mAC cells, both RAD21 alleles are tagged with AuxinInducible Degrons and an mClover reporter, and the Auxin1 gene is integrated at the AAVS1 locus. Auxin treatment leads to proteasomal degradation of RAD21. Live cell imaging after Hoechst 33342 staining to label nuclei. Nuclear mClover fluorescence corresponding to tagged RAD21 was lost after 1 hour of Auxin treatment. See SMC1 and CTCF ChIPseq signal with and without Auxin treatment. RAD21, SMC1, and CTCF ChIPseq signal left, middle, right across all peaks called for each of the proteins in untreated RAD21mAC cells. Top average enrichments for each protein. After RAD21 degradation, the cohesion complex no longer binds to chromatin. CTCF binding is unaffected. Credit cell, DOI 10.1016J, cell .2017.09.026A multi-institutional team spanning Baylor College of Medicine, Rice University, Stanford University and the Broad Institute of MIT and Harvard has created the first high-resolution 4D map of genome folding, tracking an entire human genome as it folds over time. The report, which may lead to new ways of understanding genetic diseases, appears on the cover of Cell. Making connections for decades, researchers have suspected that when a human cell responds to a stimulus, DNA elements that lie far apart in the genome quickly find one another, forming loops along the chromosome. By rearranging these DNA elements in space, the cell is able to change which genes are active. In 2014, the same team of scientists showed it was possible to map these loops. But the first maps were static, without the ability to watch the loops change. It was unclear whether, in the crowded space of the nucleus, DNA elements could find each other fast enough to control cellular responses. Before, we could make maps of how the genome folded when it was in a particular state, but the problem with a static picture is that if nothing ever changes, it's hard to figure out how things work, said Suhas Rao, first author of the new study. Our current approach is more like making a movie. We can watch folds as they disappear and reappear. One ring to rule them all to track the folding process over time. The research team began by disrupting cohesion, a ring-shaped protein complex that was located at the boundaries of nearly all known loops. In 2015, the team proposed that cohesion creates DNA loops in the cell nucleus by process of extrusion. Extrusion works like the strap length adjuster on a backpack, explained Dr. Erez Lieberman-Aden, director of the Center for Genome Architecture at Baylor College of Medicine and senior author on the new study. When you feed the strap through either side, it forms a loop. DNA seems to be doing the same thing, except that cohesion rings appear to play the role of the adjuster. Aden said a crucial prediction of the 2015 model is that all the loops should disappear in the absence of cohesion. In the new research, Aden, Rao and colleagues tested that assumption. We found that when we disrupted cohesion, thousands of loops disappeared, said Rao, a medical student at Stanford University, Hoechst fellow and member of the Aden lab. Then, when we put cohesion back, all those loops came back, often in a matter of minutes. This is precisely what you would predict from the extrusion model, and it suggests that the speed at which cohesion moves along DNA is among the fastest for any known human protein. Loops versus groups, but not everything happened as the researchers expected. In some cases, loops did the exact opposite of what the researchers anticipated. As we watched thousands of loops across the genome get weaker, we noticed a funny pattern, said Aden, also a McNair scholar, Hoechst fellow and a senior investigator at Rice University's Center for Theoretical Biological Physics. There were a few odd loops that were actually becoming stronger. Then, as we put cohesion back, most loops recovered fully, but these odd loops again did the opposite. They disappeared. By scrutinizing how the maps changed over time, the team realized that extrusion was not the only mechanism bringing DNA elements together. A second mechanism, called compartmentalization, did not involve cohesion. The second mechanism we observed is quite different from extrusion, explained Rao. Extrusion tends to bring DNA elements together two at a time, and only if they lie on the same chromosome. This other mechanism can connect big groups of elements to one another, even if they lie on different chromosomes. And it seems to be just as fast as extrusion. Broad Institute Director Eric Lander, a study co-author, said, "We're beginning to understand the rules by which DNA elements come together in the nucleus. Now that we can track the elements as they move over time, the underlying mechanisms are starting to become clearer." Explore further. Researchers identify molecular motor that transforms chromosomes. More information. Cohesin loss eliminates all loop domains. Cell. doidx.doi.org 10.1016j.cell.2017.09.026 www.cell.com Chelfultex 009286741731120 A journal reference. Cell provided by Baylor College of Medicine.